Cell Signaling, Part 3. And what we have been talking about is the G protein coupled receptor that can either activate the adenylate cyclase, and this produces either in an increase or decrease in the intracellular levels of cyclic AMP. With the increase of intracellular levels of cyclic AMP, this type of signaling can result in the activation of protein kinase A and then activation of a number of different cell signaling processes, depending on which cell type that you are looking at. So now what I want to look is the G protein coupled receptor that can activate phospholipase C, and that's shown here in the right hand side of the slide. So here you have a signaling molecule that binds to the plasma membrane receptor, and this G protein coupled receptor activates the G protein, and this is a different type of heterotrimeric G protein that can go on and activate phospholipase C or PLC. The activation of the phospholipase C result in the accumulation of different second messenger, which is IP3 and calcium. These second messengers can then go on and influence the activity of a wide variety of target proteins and often induces changes in gene expression. So let's look more specifically at IP3 and calcium, and then we'll look at the mechanism of this signaling pathway. Now here is a slide that shows some of the lipid molecules that we saw when we were discussing biomembrane structure. We also mentioned that inositol 1,4,5 phosphate or IP3 is a signaling molecule shown here at the bottom right hand side of your slide that is generated by the action of phospholipase C. Phospholipase C breaks down PIP2, which is PI4,5 bisphosphate or PIP2, into two entities. One is a diacylglycerol or DAG, which is still associated with the membrane, and the other one is the IP3 or inositol 1,4,5 triphosphate, which is commonly known as IP3. Now, what's the origin of the PIP2? We have to go back to several steps. PIP2 is made from phosphatidylinositol or PI through the action of PI4 kinase, resulting in the PI4 phosphate or PIP. Then the PIP can be phosphorylated by the PIP5 kinase to result in PIP2. And then for our discussion in signaling, PIP2 can be cleaved by phospholipase C to form diacylglycerol, which is also known as a DAG, and IP3. Now at the bottom of the slide, that what you are seeing, that how the IP3 signaling molecule is going through a dephosphorylation step to form PIP2 or PIP. This pathway is involved in reutilization and regeneration of IP3 back again so that the top portion of the figure that can go on again. So this is the reutilization pathway or the step. So what about calcium? So calcium is also a very, very important signaling molecule, and its level are kept very, very low in the cell by a number of different mechanisms. We saw one of these already when we were talking about coupled transporters, and we saw how calcium levels were maintained at very low levels in the cell through the sodium-calcium antiporter. So this is one that we have seen already in our discussion on the transport across membranes. But the cell has a wide variety of other mechanisms for keeping the levels of calcium low in the cytosol. One of them is through the active transport. So here is a well-characterized calcium ATPase, which can use the energy of ATP hydrolysis to pump the calcium from the cytosol into the extracellular space. There is also calcium ATPase that is found in the membrane of the endoplasmic reticulum, and in this case, this actively transporting pump can move calcium from cytosol into the lumen of the ER. There are a variety of calcium binding proteins in the cytosol that bind to calcium and keep calcium from propagating any kind of signal, because calcium is a very, very powerful signaling molecule. And then finally, there are a wide variety of other transporters that we are not really going to look at, but they can transport calcium from the cytosol into other organelles such as a mitochondria. The bottom line here is that the level of calcium in the cytosol of eukaryotics is quite low because it is a very powerful signaling molecule, and that eukaryotic cells through signaling can increase the levels of intracellular calcium to propagate signals. So here is how the G protein coupled receptors can now activate phospholipase C. So here you have a heterotrimeric G protein that activates adenylyl cyclase, were called the stimulatory G protein or GS, and those that inhibit the adenylyl cyclase were known as a GI for inhibitory G proteins. Now these G proteins also have the same structure. They are heterotrimeric. They do have an alpha subunit, a beta, and a gamma subunit, but they are called GQ or heterotrimeric GQ proteins. The name was quite arbitrary. Unlike S, which means stimulatory, and I means inhibitory, Q actually doesn't mean anything. It is an arbitrary designation by the cell biologist who first described this particular signaling pathway. The GQ protein works in the exact same way that we saw for the stimulatory G proteins. So on the left-hand side of your slide, C receptor is a GQ-linked receptor, and it binds to some type of extracellular signaling molecule. This activates the receptor such that it can now interact with the heterotrimeric GQ protein. In this case, when the alpha subunit exchanges GDP for GTP, it dissociates from the beta-gamma dimer, then it moves in the membrane and interacts with the phospholipase C and actually activate phospholipase C, and the active phospholipase C then cleaves PIP2 into IP3 and diacylglycerol, which is still attached with the membrane. The IP3 can then move into the ER and induce the calcium release from the endoplasmic reticulum. On the other hand, the diacylglycerol can interact with a kinase known as protein kinase C and cause activation of a variety of cellular proteins. And this activation of protein kinase C is aided by the calcium that is being released from the ER. So let's take a look at this a little bit more detail. So this part is very similar to what we have seen in the previous slide. So let's start here. So here is PIP2 in the membrane. Next to it is this large yellow transmembrane protein is active phospholipase C, which can cleave PIP2 into IP3 and DAG. Then IP3 moves to the endoplasmic reticulum and activates the IP3 gated calcium channels. These calcium channels open up and the calcium flows from the lumen of the ER into the cytosol. On the other hand, the diacylglycerol, which is still associated with the plasma membrane, will interact with the protein kinase C. And with the help of a calcium that is being released from the ER, the protein kinase C can go on and phosphorylate a wide variety of substrate, which we are going to see in a minute. And the outcome of that is most often is a change in gene expression. The other consequences is what happens if all these stored calcium is used up from the ER and if the cell requires more. So we're going to talk about that, what happens in this condition in the step 8, which is in the next slide. So here in this picture, we are looking at a plasma membrane associated calcium channel, which is known as an ORI1, which is unlike any other transport channel and is very unique. And the other protein is a calcium sensing protein known as a STIM1, STIM for stromal interaction molecule. And this is an ER membrane associated protein or a transmembrane protein. And this can bind to the calcium, which is in the ER lumen. And there is a cytosolic domain, which is known as a CAD, and CAD stands for the calcium release activated channel activated domain, or CRAC activated domain. So on the left side, what we are seeing that at this condition, there is a high level of calcium in the ER lumen. Now the calcium is bound to the ER sensing protein, this STIM1, and it is in the native dimeric form and does not interact with the plasma membrane associated ORI1 channel. On the right-hand side, as a consequence of IP3 signaling, the ER stored calcium will move out into the cytosol and engage in different functions. We have seen the mechanism in the last slide. So the ER calcium store gets depleted and the STIM1 will also lose the calcium. And this triggers of this oligomerization of multiple STIM1 dimers and a drastic conformational change. And then the STIM1 is relocated into the ER membrane, which is close to the plasma membrane. And then this oligomeric STIM1 interacts with the ORI1 and opens up the calcium channel. So this is the consequence of IP3 signaling, that in need, the calcium can be supplied from the extracellular environment. Then once the calcium requirement is over in the cell, the calcium is either returned to the ER by calcium pump or transported into the extracellular matrix by either calcium pumps or by sodium calcium antiport that we have seen just a few slides back. On the left-hand side, what we are seeing is an inactive protein kinase C, or in this case, it's called a C kinase. It is the same thing. You have an inactive MAP kinase in green in the cytosol, and then you also do have an inactive NF-kappa B, which is associated with the I-kappa B complex and is a dimeric form. So the NF-kappa B is a transcription factor, but when it is dimerized with this I-kappa B in the cytosol, it is inactive. So if we move over to the right-hand side of the slide, now you have the appearance of the diacylglycerol in the plasma membrane through the GQ-coupled receptor signaling pathway. And diacylglycerol then can bind to the protein kinase C and activates protein kinase C, which then phosphorylates the MAP kinase, which is itself one of those relay proteins that becomes activated when it is phosphorylated. MAP kinase can move into the nucleus and can phosphorylate a wide variety of transcription factor and induce expression of a variety of genes. The protein kinase C can also phosphorylate the I-kappa B. This causes a conformational change. So NF-kappa B is then released from the I-kappa B binding partner. So then the NF-kappa B also moves into the nucleus and itself behave as a transcription factor, binding to upstream regions of genes and inducing the expression of messenger RNA, which will then eventually lead to increase in translation of proteins. The upstream regions that these transcription factors bind to, for example, this transcription factor here, and also the NF-kappa B, these regions of the DNA they bind to, they are often referred to as a serum response element. And this is actually just a historical term. If you take a mammalian cell line and you expose it to the serum, you can induce this signaling pathway and you can induce expression of genes. So that's why these genes were said to be controlled by the serum response element, or SRE. Okay, so that concludes our discussion on the G protein coupled receptors, or G protein linked receptors. And we saw three different protein receptors: those that bind to the stimulatory G proteins, those that bind to inhibitory G proteins, and those bind to the G proteins that activate phospholipase C. So these were the three examples of the G protein linked receptors we looked at. Now I want to turn to the enzyme linked receptors. So enzyme linked receptors are so named because they themselves have a catalytic capability. They themselves are enzymes, and in their inactive state, or when they are not bound to the ligand, their catalytic domain is inactive. And when they're bound to the ligand, their catalytic domain, usually on the intracellular side of the plasma membrane, becomes activated. The enzyme-linked cell surface receptors are a very large family. They are receptors guanylyl cyclase, and these are receptors that once bound to the ligand can actually convert GTP to cyclic GMP. And very similar to the adenylyl cyclase, can convert ATP to cyclic AMP. Probably the largest subfamily of the enzyme-linked cell surface receptors are the receptor tyrosine kinases, and I have listed a number of them here: nerve growth factor, platelet-derived growth factor, fibroblast growth factor, EGF receptor, VEGF receptor. These are all plasma membrane proteins that have the capability of phosphorylating tyrosine residues, hence their name is tyrosine kinases. There are the receptors tyrosine phosphatases, and as the name suggests, they are cell surface receptors that can dephosphorylate the phosphorylated tyrosines. And then there are receptors, those are serine threonine kinases, and these are receptors that can go on phosphorylate serine and threonine residues in other proteins. So for our discussion, we are only going to focus on the receptor tyrosine kinases. And you may have heard about the EGF receptors and the VEGF receptors because these are the receptors that are often associated with cancers. They are often mutated in cancers. The EGF receptors are often mutated in the breast cancers, and the VEGF receptors can participate in the growth of blood vessels towards tumors. And so tumor cells really rely on VEGF and the VEGF receptors to get nutrients. And we are going to discuss this a little bit more when we talk about the cancer cell biology at the end of this course. But today we are just going to look at the receptor tyrosine kinase in general. Okay, so here is a figure that illustrates the structure and activation of these receptor tyrosine kinases. So all these receptor tyrosine kinases have a very similar structure. They have a large extracellular domain that can bind to a ligand, and they have this transmembrane alpha helix, and they have a cytosolic domain, and it is the cytosolic domain that you have the tyrosine kinase catalytic domain. So the tyrosine kinase activity is associated with the cytosolic domain. And when these cell surface receptors are not activated, so when they are not bound with a ligand, they exist in monomers in the plasma membrane. So they are single proteins in the plasma membrane. However, when these receptors actually encounter and bind a ligand, they dimerize, as shown in this step 2. And it's the dimerization event that activates the tyrosine kinase's catalytic domain, and you see phosphorylation. And I should say that the tyrosine kinase domain actually phosphorylates tyrosine kinases in the receptor cytosolic domain itself. So it's actually an autophosphorylation event. So the receptor tyrosine kinase, once activated, it phosphorylates its own tyrosines. So you can see in the step 2, you have phosphorylation. You have the beginning of phosphorylation of the catalytic domain. And then as the activity increases, you get multiple phosphorylation events on the catalytic domain. So this is the first step of the activation of the receptor tyrosine kinase. The next step involved the G protein. So one of these relay proteins that we have also seen before, and this protein is RAS. And you will remember that we talked about RAS before, and I mentioned that the RAS is a molecular switch because like all GTP binding relay proteins, it is inactive when it's bound to GDP, and it is active when it is bound to GTP. And this exchange of GDP and GTP is facilitated by a large class of protein, which is known as the guanine nucleotide exchange factor, or GEF. Sometimes they are called GNRP, guanine nucleotide releasing protein, or GNRF, like guanine nucleotide releasing factor, etc. But the more common name is the GEF, which stands for the guanine nucleotide exchange factor. And so it's this protein that facilitates the exchange of GDP for GTP and activation of the RAS protein. Once the RAS propagates its signal, it needs to be turned off. And so the intrinsic GTPase activity of the RAS itself can hydrolyze GTP to GDP and turn itself off. But the intrinsic GTPase activity of the RAS is quite low. And so it needs the help of another protein, which is known as GAP, which stands for GTPase activating protein. But this is very important. It is not the GAP that is hydrolyzing the GTP to GDP. It is RAS itself. The GAP is only helping to enhance the intrinsic activity of the RAS, but all GTPase activity is in the RAS itself. Okay, so here is the continuation of the activation of this receptor tyrosine kinases. So let's start from the beginning. This is the upper left-hand side of the slide. Here is an inactive tyrosine kinase existing as a monomer. Binding of ligand that results in dimerization of the receptor. which then results in autophosphorylation of the cytoplasmic domain of the receptor. The next protein to bind, which is shown in the upper right-hand side of the slide, is a protein known as GRB2. And this is a very interesting protein because it has two very specialized protein domains. It has an SH2 domain, which can bind to the phosphorylated tyrosine. And it's no surprise that it binds to the extensively phosphorylated cytoplasmic domain of the receptor tyrosine kinase. It also has an SH3 domain, and these are specialized protein domains that can bind to the proline-rich region of other proteins. So here is the SH3 protein that recruits a second protein known as SOS. And as it turns out, SOS is a RAS GEF, so SOS can then bind to inactive RAS. So here you can see in light blue the inactive RAS bound to the GDP. So SOS acting as a guanine nucleotide exchange factor that can facilitate the exchange of the GDP for GTP, thus activating RAS. So here are the steps once again. You have the binding of the ligand, dimerization of the receptor, then you have the autophosphorylation of the tyrosine residues, recruitment of GRB2, recruitment of SOS. SOS is a GF that then recruits RAS, and the SOS facilitates the exchange of the GDP for the GTP, thus activating the RAS. So one of the questions that you might have is how quickly does RAS become activated? And this is a question that we have addressed previously when we were looking at methods in cell biology. And again, we are turning to this powerful technique of FRET, fluorescence resonance energy transfer. And again, I remind you that this is a powerful fluorescence technique in which you can tag the two entities in the cell with the fluorophores, and as long as the fluorophores are a good FRET pair, in other words, the emission wavelength of one of the fluorophore, the donor fluorophore, is the excitation wavelength of the acceptor fluorophore, you can use this to determine how close two entities are in the cell. And in our particular discussion, you can use this to follow the RAS activation in real time to see how quickly RAS can be activated by these receptor tyrosine kinases. So we have already seen that the G protein of the RAS family cycle between the GDP-bound inactive and GTP-bound active forms. The cycle is regulated by the GEF, the guanine nucleotide exchange factor, the activator, and the GAP, the GTP is activating protein, the inactivator. Now, 20 years ago, a group of Japanese scientists at Osaka University carried out an experiment to follow the RAS activation in real time and published in Nature in 2001. They asked the question, where in the cell RAS get activated and how fast? They answered the question using FRET. So the scientists designed a complex chimeric protein with a YFP or yellow fluorescent protein on the N-terminus, followed by a RAS. Then you have a flexible linker, and then you have a protein RAF, which only binds to RAS in a GTP bound state. And then you have a CFP or cyan fluorescent protein. The scientists transfected some COS1 cells in the culture to produce this chimeric protein. So when there is no signaling through the receptor tyrosine kinase, the RAS is in the inactive GDP bound state, and the RAF does not bind to RAS. In this case, the YFP and the CFP are far apart and there is no FRET. If you excite the cells at the excitation wavelength of the CFP at 433 nanometer, you only get the emission spectrum of 475 nanometer of CFP. Now, the scientists added some EGF or the epidermal growth factor to the cells, which signals through the receptor tyrosine kinase. The activation of the receptor tyrosine kinase activated the guanine nucleotide exchange factor or GF, and upon the exchange of the RAS GDP, now it becomes RAS GTP. This form of RAS will immediately bind to RAF, and they are now close and within 10 nanometer distance. So the YFP and CFP will show FRET. So when the cells are excited at 433 nanometer, CFP will emit 475 nanometer, which is the excitation wavelength of the YFP, and YFP being closed due to FRET will emit at 527 nanometer. So here it is how it looks like under a fluorescence microscope. So EGF was added to some COS1 cells in culture, which activated the receptor tyrosine kinase. And you can see within about 5 minutes, the accumulation of this red-orange fluorescence around the periphery of the cell. And by about 30 minutes, you can see that RAS is significantly activated because you have such an abundance of this red-orange fluorescence, which suggests that you are getting a FRET signals between the RAS-YFP and the RAF-CFP. So the images are actually stacked and so the video as well. So let's take a look at this video that you are seeing the cell in the phase contrast at the upper left, CFP at the lower left, and the YFP at the lower right were obtained every 20 seconds and the images were stacked, and that's the FRET signal. So that in real time, you can see that what it is. Now, another important thing that is important that you are seeing that we are talking about this YFP or the yellow fluorescence protein, but it is green. But the scientist actually particularly said in the paper that it is a green fluorescence protein which has been mutated, and they named it a yellow fluorescence protein, but it is not really. The emission spectrum doesn't again show that. And here you can see the FRET pair that when they were close enough, that right here, this is the FRET signal at 527 nanometer. That's what you are observing in your picture. So now let's take a look at that. What happens to downstream of RAS? So once you have activated RAS in the membrane, which is in the step 1 in this slide, then active RAS then can recruit and bind another kinase known as RAF. Now, the RAF is normally in an inactive state in the cytosol, as shown in here, and it is bound with an inhibitory protein called as 1433. Once the RAF binds to RAS, then two things happen. First, you have the release of the 1433 protein, which is not shown in the slide, and then you have the deactivation of RAS by the hydrolysis of GTP to GDP. And after you have the inactivation of RAS, then you get dissociation of RAF kinase from the RAS. But now RAF is in its active state. It has undergone some autophosphorylation, so it has picked up some phosphate groups, which helps in its activation. Now it has lost its inhibitory protein 1433, so RAF can then go on and phosphorylate MEK, which is another kinase, and so it activates that kinase. And then the MEK can phosphorylate the MAP kinase, which activates the MAP kinase. So the MAP kinase regulates the activity of many transcription factor, controlling approximately 1100 early response genes activated by adding a growth factor to quiescent cells. And that's why we call it as a mitogen activated protein kinase or MAP kinase. So what happens downstream of MAP kinase? Two things happens together. In the first step, 1 through 3, MAP kinase is in the cytosol and phosphorylates and activates this P90 RSK. And the RSK stands for ribosomal S6 kinase, which is a family of kinase. So this is your P90 phosphorylated P90 RSK kinase. The active P90 then moves into the nucleus and phosphorylates SRF, which is a serum response factor, which is a transcription factor. Now, second, in steps 4 and 5, MAP kinase translocates into the nucleus and phosphorylates this TCF, which is the ternary complex factor. This is a transcription factor already bound to the promoter of the CFOS gene. And at this point, the phosphorylated TCF and SRF together stimulate the transcription of the CFOS and other genes that contain a promoter of the SRE or the serum response element. MAP kinase phosphorylates over about 175 target proteins in the nucleus, so this is just one of them. Now, we talked about the MAP kinase earlier, and we are talking about the GQ coupled protein response, but I don't want you to get an idea that the MAP kinase is just one type of enzyme. In fact, there are dozens of different MAP kinases, and this figure illustrates this. This is a series of MAP kinases that are found in yeast and illustrating the outcome of signaling from those MAP kinases. And these outcomes include mating, then filamentation, osmolyte synthesis, cell wall remodeling, and sporulation. Here are the types of signal that activates these MAP kinases. For example, the pheromones, starvation, high osmolarity, hypotonic shock, carbon and nitrogen deprivation. So the whole reason that I'm showing this figure is just to have you an idea that the MAP kinases are very large family of signaling molecules, and they can behave in many different ways in different cells. I just want to give you an idea that MAP kinase is not a single protein, but a large number of proteins, which can elicit a lot of different responses in the cells. So up to now, we have been looking at examples of signaling within the cells, and we have been talking about what turns on these signals in the cell. But a discussion of cell signaling is incomplete unless we talk about how cells may turn down this type of signaling. So I just want to very briefly in general look at two different ways in which cells, the target cells, actually adapt to signals and actually turn down signaling once it is activated. One of these happens very slowly. It is called the adaptation or slow adaptation. We're actually seen this already, but we call this as receptor downregulation or receptor mediated endocytosis. And I will show you in a slide in a minute that illustrates the slow adaptation or receptor downregulation. And hopefully you will recognize that from a previous lecture. And then there is a rapid adaptation, which can come from the receptor phosphorylation, and we will take a look at a couple of these examples. Okay, so slow adaptation is also known as receptor downregulation, and this is when a receptor is internalized through the receptor-mediated endocytosis, and that stops the signal. So here is a cell, and it has on its cell surface a receptor for epidermal growth factor or EGF receptor. And while that signaling molecule is bound to the cell surface receptor, a signal is being propagated, so through RAS, RAF, MEK, and MAP kinase, etc. Now, you will remember that when we talked about the receptor-mediated endocytosis, and that this occurs at a very specialized domains in the membrane known as clathrin-coated pits. And clathrin-coated pits have the propensity to attract the receptors. So if this EGF-bound receptor happens to find itself in the clathrin-coated pit, it will be endocytosed by the receptor-mediated endocytosis. And then probably this receptor ligand complex will be trafficked to the lysosome via endosome and will be degraded. The ligand will be degraded and the receptor probably will also get degraded. So in this way, the cell has turned off the signaling from the EGF receptor. It is called a slow adaptation because it is not regulated. The receptor and the ligand have to just find themselves in clathrin-coated pit by chance, and that's why it is referred to as the slow adaptation. So the phosphorylation of a receptor is a much faster way for the cells to stop signaling. So for example, the receptor for adrenaline, the GS alpha subunit, which in turn activates cyclic AMP, which then turn activates protein kinase A. But what is interesting is that the protein kinase A can then come back and phosphorylate this receptor and turn it off. And this can happen pretty quickly. The other receptors can be phosphorylated by other kinases in the cell, and the phosphorylated receptor can bind to other proteins and can block signal. So for example, here is your activated receptor. It becomes phosphorylated, so it becomes inactive. And so now it has a number of these phosphorylated amino acid, and these phosphorylated amino acids can bind to other proteins, an example of which is a beta-arrestin, which can block further signaling from the cell. And this concludes the part 3 of the cell signaling discussion.